Hello students, today I am here to give you and to teach you one topic and from the chapter that is cell cycle and cell division. We know that cell is a very important part of our body and it plays one of the most striking characters in our body, that is it provides the structure. That's why it is also known as a fundamental unit of life. Now, first of all we need to know why the cell is called the fundamental unit of life, because cell performs all the necessary functions of an human or any living organisms. Okay, all the necessary functions to survive in this planet. Whatever the necessary functions, what are the metabolic activities we need to perform, all the chemical reactions are performed by the cell. That is why cell is known as the fundamental unit of life. Fundamental unit of life. Okay, cell is also known as the structural unit of life. Why the cell is known as a structural unit of life? You see, because in living organisms all the organs, tissue, all the components of the body are made up of cell only. That is why cell is known as a structural unit of life. So generally we need to in our topic we need to understand two types of cell division are there. We know that is the mitosis and the meiosis. So cell division can be of two types. One is mitosis, another one is meiosis. Are you getting my point? So mitosis is given by Fleming, observed by first of all Fleming, and meiosis Murray and Farmer. These are the name of the scientists who have termed the meiosis cell division, and Fleming has given the mitosis cell division. Are you getting my point? And we know that our body, there are two types of organisms. No, one is a unicellular, for suppose unicellular. What is unicellular organism? We know. One is a multicellular organism. So multicellular organism, their body is made up of many cells. Okay, and unicellular organism, their body is made up of only one cell. We know. That means in unicellular organism, single cell can perform all type of functions. Either it can be respiration, it can be excretion, it can be transportation, all the necessary functions that we need to survive. All the functions can be performed by a single cell. That they can perform all the functions only with the single cells, though they are unicellular organism. And these are the multicellular organism. We know we have the special structures to perform different type of organisms. Simple organism doesn't need that type of structures. Their single cell can perform all the functions. So in unicellular and multicellular, okay, we know all these things, but in the multicellular organisms, many cells combines. What it forms? It forms tissues, and this tissue will form organ, and this organ will form organ system. Are you getting my point? That means cell needs to divide. Our body consists of many cells. Are you getting my point? But during reproduction, okay, how we came, when the sperms and ova fertilized, then only we were produced. There is a zygote. Sperm and ova fertilized, we will study in the later chapters. Sperm and ova will get fused. Okay, they will fuse together and they will form what? They will form zygote. Now you see, zygote is a single cell. After that, 9 months of pregnancy will occur. After that, a baby will be formed. But you see, the baby body consists of billions of cells. Are you getting my point? Because baby body contains of organs, tissues, muscles, many things are there. Their body are made up of billions of cells. Now the question is, from zygote that is formed after the fusion of sperm and ova. Are you getting my point? Now, how that baby, after 9 months, that baby contains of billions of cells. From single cell, how we are able to produce an individual which contains many cells. That means how the cell has performed its division, how the cell has produced from one cell many cells. Okay, and this phenomena is known as cell division. Through the process of cell division, one cell will divide and it will form many cells. Okay, cell arises from where? Cell arises. Cell comes from the pre-existing cells. Cell arises from pre-existing cells. Now this statement, cells arise from pre-existing cell. That means one cell arises from another cells. Okay? So this statement is given by Rudolf, one scientist whose name is Rudolf Virchow. Rudolf Virchow. This statement is given by him. Are you getting my point? Okay. Cell arises from pre-existing cell. Exactly statement, if you want to know, then it is omnis cellula a cellula. Okay, it is given in our NCERT book, omnis cellula cellula. What does it mean? It means cell arises from the pre-existing cells. Okay, given by the Rudolf Virchow. Okay, somebody has asked that we know that cell, living organism contains cells. Cell are made up of cytoplasm, cell are made up of cell membrane, cell are made up of cell wall. Okay, all the things, these are the constituents of the cell. But the question is, from where the cell arises? Cell arises from the pre-existing cells. Okay, one cell will divide. What this statement, what does it mean? That means one cell will divide and it will form two cells. Now this is known as the cell division. This is known as the cell division. So this is the topic I am trying to make you understand. There is a cell division, how cell will divide and how it will produce another cell. Now the thing is, you see, cell will undergo one cycle. For cell division cell will undergo one cycle. That cycle is known as cell division. First stage is a G1 stage, second stage it is a S stage, third stage it is a G2 stage. Okay, this one is known as the M phase or the mitotic phase, and this stage is called cytokinesis. Are you getting my point? So from here to here, this stage is the longest phase. Okay, and it is known as the interphase. Interphase. Okay, so this is known as the interphase. In this stage, all the necessary things that is required to make one cell. Suppose you say this is a cell. What does it contain? It contains cell wall, it contains cell membrane, all of these things we know na. Cell contains what? Cell contains, if it is plant cell, then it is made up of cell wall. Some microorganisms like bacteria, their cell also have cell wall. It has what? It has plasma membrane. Okay, so this is what? This is a plasma membrane. Cell contains nucleus. We know cells contain nucleus. Now if these cells wants to form another new cells, suppose another new cells, okay, so it needs to produce cell wall, it needs to produce cell membrane, it needs to produce nucleus, and inside the nucleus what we have? Inside the nucleus we have chromosomes. Now if this cell wants to divide, first of all it needs to produce all these materials, then only it will be able to divide, because the new cell that will be produced, it should contain all the materials, na? All the materials of a cell. So this thing will be produced, these things will be produced in these phases, that is the interphases. To produce one nucleus contains what? Chromosomes. To produce new chromosomes, suppose in case of us, what is the number of chromosomes? 46. To produce one new cell, we need another copy of 46. Is it correct? To produce another cell, we need another copy of 46. To produce chromosomes, we need DNA. To produce DNA, we need nucleotides. To produce nucleotides, we need many bases, nitrogenous base pairs, then phosphorus, then sugar. All these things have to be present. So in these stages, that is this phase, there is a G1 phase, S phase and G2 phase. In this phase, all the things that are necessary to produce this cellular organelle, it can be nucleus, it can be chromosomes, to produce new nucleus, to produce new mitochondria, to produce new Golgi apparatus, okay, to produce endoplasmic reticulum, to produce new endoplasmic reticulum. These are known as what? These are known as cellular organelles, organs of the cell. These are also present here. Now this cell, if needs to divide, it needs to produce another copy of these cellular organelles, but for the preparation we need some raw materials. So these raw materials, they will produce here. These raw materials, they will produce here. Are you getting my point? So this phase is the longest phase. It is known as the interphase. It consists of 95% of the cell division. 95% of the cell division is taken by the interphase only. This phase, that is the M phase. Okay, this phase. In this phase, only 5% of the time have been consumed. 5% of the time. It takes for 24 hours. It takes 24 hours for a human RBC cell to divide. But in case of yeast, it contains only 90 minutes. It takes only how much? Yes, 90 minutes, 1 hour 30 minutes. Okay, but in case of the human RBC, it will take 24 hours. So in case of the 24 hours, if you find out the 95% time, most of the time of the 24 hours, it's taken by the interphase stage. Maximum stage will be taken by the interphase stage, because for the preparation of all the cellular organelles, they need time. Okay, and only cell division will occur only in the M phase. Cell division will occur only in the M phase. When the preparation is done, suppose for the if we go for cooking of food, it takes time. But when we eat that material, it takes very less time. So the thing is like that. If we all the necessary materials, to produce all the necessary materials, we need time. That is the 95% of the time. From 24 hours, 23 hours will be taken up by the interphase, and actual cell division will take place only in this phase. That is the M phase, which needs only 1 hour. Are you getting my point? Now you see this G1 phase, the time taken by from this 24 hours, maximum time. Okay, in case of human being, it's 24 hours. So in case of animals, in case of any animals or any plant cell, whatever the time it's there, whatever the time they need to perform these whole stages, many stages, there is a G1, S, G2 to M, togetherly they are known as what? They are known as a cell cycle. They are known as cell cycle. So to perform cell cycle by the organisms, whatever the time they need, 42% of the time is taken by G1 phase. 35% of the time is taken by the S phase. G2, it contains many less time than 35. It will be 21.2. Are getting my point? And less time is taken by, as we know, mitosis. Only where the cell division, actual cell division will take place. Actual cell division will take place in the mitosis, and it takes less time than the G2 phase. Maximum time is taken by the G1 phase. Now, what is G1? G1 means what? Okay, suppose here G1 phase means gap phase. Okay, S means synthetic phase. Okay, now in gap phase, what are the things that is produced? In G1 phase, it will produce proteins. Now for proteins, how the proteins are formed? Proteins are formed by one process. It is known as the translation. So in G1 phase, what will happen? Protein will take place. Translation will be there. Translation, transcription. Okay. How the proteins are produced? Proteins are produced by these two processes, transcription and translation. So what I have told you? I have told you that in G1 phase, there is a gap phase 1, proteins will be produced. So to make proteins, obviously this process will be there in the G1 phase. In S phase means synthetic phase. What are produced in S phase? In S phase, you see, DNA replication will take place. DNA replication. We know what is DNA replication. DNA replication means a duplication of DNA. From one DNA, another DNA will be produced. No. So for DNA replication, we need one protein. We know what is the protein. The protein is a histone protein. Because DNA are packed with the help of the histone protein. So if DNA replication will take place in S phase or synthetic phase, histone protein will be also produced there. So you can remember like this: in S phase, DNA replication and histone protein are produced. Another thing that is produced in the S phase or synthetic phase is the formation of the centriole. That is the centriole duplication. Now the centriole duplication will start. Centriole duplication starts in the S phase. So in S phase, DNA replication has taken place, histone protein is produced, and centriole duplication started. Now G2 phase. What will happen in the G2 phase? Okay. What are the things that are produced in the G2 phase? In G2 phase, this centriole duplication, that is the centriole duplication completes. Here the centriole duplication completes. Okay, finished. Okay, and RNA are produced. RNA produced, protein produced. We know that to make protein, RNA is required. So RNA will be produced in the G2 phase along with the RNA. So RNA produced, that means protein will also get produced. That means in G1 phase, proteins, okay, RNA are also produced here. RNA are also produced here. Are you getting my point? Nucleotides are also produced here. Nucleotides are also produced. Okay, nucleotides are also produced. So what we came to know, that in G1 phase, nucleotides, RNA, protein, translation, this is the process through which the protein will be produced. Translation, transcription will be there, and RNA, nucleotides and many other things will get produced. This is the main thing we need to remember. Okay, it will ask in the CBSE as well as in the Neet. Okay, very important. In S phase, what are produced? DNA replication. You see, in S phase, DNA replication will take place. For DNA replication, what we need? Nucleotides. So nucleotide have to be produced in the earlier phase. So nucleotides is produced in the G1 phase, and in the S phase, with the help of these nucleotides, with the help of this RNA, with the help of these proteins, DNA replication has taken place. Are you getting my point? Now, from one DNA, another DNA is produced. Now, it needs packaging. We know DNA packaging. For DNA packaging, we need histone. So, histone protein will also produce in the S phase. Next phase is the G2 phase. In G2 phase, what will happen? The duplication of the centriole. Duplication of the centriole that has taken, that is started in the S phase, as I have written here, you can see it will get completed here, and RNA protein same will be produced in the G2 phase. Another phase is there which is known as a G0 phase. G0 phase. Now you see the cell will enter in the G1 phase, next it will go to the S phase, next it will go to the G2 phase. By passing all these stages, if all the things are done, all the necessary things are produced, now they will go for the division in the M phase. M phase is known as a mitosis phase. Okay, M phase. Okay, M phase is known as the mitosis phase, where the actual cell division will take place. Actual cell division means the division of the nucleus. So either they should enter, either after performing M phase, after performing G1, S, G2, M phase, either now next time they need to go, whatever the cell is produced, they need to go to G1 phase or not. It will get dependent. There are some checkpoints, there are some proteins which are controlling these phases, which will tell the cells that yes, you need to divide. If you don't need to divide further, this checkpoint, for example, you say cyclin dependent protein, cyclin dependent protein. Okay, cyclin dependent protein. Then what you can say, platelet derived factor, platelet derived factor, epidermal derived factor. Now you see these things will encourage the cell. These things will encourage the cell to divide. These things will encourage the cell to divide. Now after passing G1, S, G2, M, cytokinesis, cell is produced from one cell in case of mitosis. What we are discussing, I am telling you the mitosis. Now this cycle G1, S, G2 will also, they will come under mitosis also and meiosis also. But you see, in mitosis from one cell 2 cells are produced, and meiosis from one cell 4 cells are produced. So either they should go further. Suppose there is one cell, it has prepared all its raw material, and it has went to perform mitosis, it has went to perform cytokinesis, all things are done. From one cell 2 cells are produced. Now these 2 cells that is produced here, suppose these 2 cells that is produced after going through the M phase, G1 phase, S phase, all these phases, these 2 cells, they need to divide further or not. It will be guided by these factors, platelet derived factors, epidermal derived factors. This factor will encourage to go for cell division. Are you getting my point? But you see, sometimes they are also told so not to divide. They are also told not to divide. So these cells, that is the new cell that is produced, will enter in the GO phase. If the cells, these 2 cells doesn't divide, okay, then they will go under GO phase. That doesn't mean that they are not capable to divide. They are able to divide, but they will divide when there is a requirement. Our body, our cell will told them Okay, they will tell them when to divide. When our body or cell will tell them to divide, then only they will go for division. Otherwise, they will not go for division. These two cells or any cells, if they go to the geo phase, if they don't divide, they are metabolically active. What does it mean? That means they can divide. They will divide when they are told so, and they will not divide if they remain in the geo phase. Getting my point? Okay. Next is the M phase. So after preparation of all the raw materials, the material that we need, that are needed for the cell division, okay, all these things are produced. Now the cell division should take place. Now they will enter M phase. Now the actual cell division takes place. You see, in M phase what happens? Karyokinesis. You see, cell division, during cell division, excuse me, during cell division, one stage is known as a mitosis phase. In mitosis phase, it is just karyokinesis. Karyokinesis. Another one is the cytokinesis. What will happen in the karyokinesis? In karyokinesis, the nucleus will divide. That means this is the cell, this is the nucleus. From one nucleus, another nucleus will be formed. I have told you inside the nucleus what is present? Chromosomes. Suppose in case of the human being, how many chromosomes are produced? 46. So from 46 chromosome, another 46 chromosome will be produced. Where? In which stage? In this stage. There is a karyokinesis. We have entered in the M phase. After crossing G1, S, G2, all the necessary materials are produced. Now we have entered in the M phase. This M phase contains two stages. Number one, karyokinesis. Number two, here I have written cytokinesis. In karyokinesis, the division of the nucleus will take place. In cytokinesis, the cell division will take place. In karyokinesis, division of the nucleus will take place. So I am writing here, you see, in karyokinesis, the division of nucleus will take place. Division of nucleus. Okay, in karyokinesis. In cytokinesis, what will happen? In cytokinesis, division of cytoplasm. Division of cytoplasm. Are getting my point? Division of cytoplasm. So first of all, we need to go for karyokinesis. In karyokinesis, there are some stages. For example, prophase. Number one, prophase. Number two, metaphase. Number three, anaphase. Okay, and last one is known as telophase. Are you getting my point? So number one stage is what? Number one stage is prophase. So you see what will happen in the prophase. Suppose you say this is the cell. Okay, this is the cell and this is the nucleus. In prophase, number one thing that will occur, it is that this nucleus has nuclear membrane. We know that nucleus has nuclear membrane. Nuclear membrane disappears. Nuclear membrane will disappear. That means it will be like this. Disappearance of the nuclear membrane will take place. Always remember, in nucleus, it contains nuclear membrane. We know nuclear membrane will disintegrate, and the nuclear membrane also has one structure. We know na, the dark structure. This dark structure is known as a nucleolus. It is known as a nucleolus. So nucleolus disappears. Nucleolus also disappears. And you see, in that stage, the chromosome will be present in the thread-like structures. So nucleolus will get disappear. It will be like this. The nucleolus has disappeared. Nuclear membrane is disappearing. Nucleolus has disappeared. This is what? This is known as a chromatin fiber. Okay, this is known as chromatin fiber. This is a chromatin fiber. This chromatin fiber is very thin, thread-like structure. So number three, what will happen? This chromatin fiber will become condensed. They will condense, become thick. These chromatin fibers, they will start to get fragmented. They will get fragmented. Small pieces. You can see small pieces. They from thin thread-like fiber, they are now condensing and they are becoming dense. They are becoming thick. So these are the changes you will observe in prophase. And Prophase at some stages, it is early prophase. Prophase has one stage which is known as early prophase. In early prophase, suppose if I write here, in early prophase stage, it has spireme stages. Spireme stage. Are you getting my point? Where spiralization of chromatin fiber will take place. Chromatin fiber will take place. Okay, it is known as a spireme stage. That means the chromatin fiber will spiralization from they are coiled now. They will uncoil themselves and they will get fragmented. Are you getting my point? So this is known as spiralization. This stage is known as a spireme stage. So all these things you will observe in the prophase. And the two nuclear centrioles. Yes, I forgot to tell you the two centrioles. These two are the centrioles. Now these two centrioles will move towards the poles. Okay, one towards the north pole, one towards the south pole. They will start movement. So centriole moves towards the poles. All these things will happen in prophase. Next stage, prophase is done. Number 2, metaphase. So in metaphase, what will happen? Suppose you see, this is a cell. As there is no nuclear membrane, all the fragments, all the chromosomes that are produced, okay, these are fragmented. It will it will look like this. All the chromatin fiber have condensed, dense, become thick, and they will appear like this. So these are the, what you can say? These are, you can say, chromosomes. It has how many chromatid? 1 chromatid. How many chromatid? 1 chromatid. So 1 chromatid. Okay. Now there, they will come towards each other. They have duplication. Duplication has been taken place. Are you getting my point? Duplication has taken place, and as there is no nuclear membrane, they are just loitering. They are just moving here and there in the cytoplasm. Earlier they were restricted to this place only, but now there is no nuclear membrane. They are moving here and there. Okay, this is what you see. When the chromatin fiber has made, this is the chromatid. Okay, this is a chromosome. You can say this is a chromosome. This is a how many chromosome? 1 chromosome. How many chromatid? 1 chromatid. How many chromatid? 1 chromatid. I have make you understand during S phase. Suppose this is a cell cycle. This is the G2. This is the S. This is the S. This is the G1, G2, S. This is the M phase. You can say no cytokinesis. M phase. So during S phase, DNA replication has taken place. I have told you. DNA replication means from 46 chromosomes. The number of chromosomes in S phase, after completing S phase, in S phase the DNA replication will take place, but the number of chromosomes will not get increased. That means the number of chromosomes in case of human being it is 46. After passing the S phase, the number of chromosomes will be same, that is a 46, remain same. But you remember, DNA has chromosome contains what? DNA. So inside the chromosome the content of DNA gets doubled. That means suppose if I write n for the number of chromosome. So number of chromosome is 46 in S phase. After S phase, in G2 phase, number of chromosome remains same, that is a 46. But the number of content of DNA, suppose in S phase, content of DNA is what? Suppose you can say twice n, or you can say 2c. Content of DNA is 2c. After the S phase, I have told you in S phase the DNA replication will take place, duplication of the DNA. So S phase it is 2c. After passing S phase, it will become, the content of DNA will become 4c, double. The content of DNA will get double. But the number of chromosome will remain same. Remember this thing. Chromosome will become dense. In S phase, suppose the chromosome is like this. After passing S phase, the chromosome will become dense. Number of the content of DNA has increased, but the number of chromosome will remain same. That's why after S phase, in G2 phase also, it will remain the same. Number of chromosome 46, content of DNA 4c. In M phase also, it will remain same. Number of number of chromosome 46 and content of DNA 4C. But remember, what happens in the M phase? What happens in the M phase? In M phase, the division, karyokinesis. In M phase, the karyokinesis, the division of the nucleus will take place. Now, this chromosome will get divided. This chromosome will get divided. So, what we are going through? We are performing the division. Prophase is done. Now, we are in metaphase. After M phase, the content of DNA will get half. Are you getting my point? In case of meiosis, it will get half. But in case of mitosis, it will be same. Number of chromosome will same. But the content of DNA will change, because you see in G1 phase, let me write here. In G1 phase, the number of chromosome is how much? 46. And content of DNA is how much? 2C in G1 phase. In S phase, number of chromosome same, 46 in S phase, 46. But the content of DNA, as DNA replication has taken place, it will be double. From 2C it will become 4C. Suppose in G2 phase, number of chromosome will remain same, that is the 46. But the content of DNA will remain same, because chromosome has become denser, but the division has not taken place. Division will take place in the M phase. So G2 phase, content of DNA double. In M phase has many stages: prophase, metaphase, anaphase, telophase. I have not written here. So in prophase, metaphase, it will remain same. In M phase, in prophase, it will remain same. That means the quantity in prophase. Okay, let me write here. Prophase, the number of chromosome will remain same, 46. But the content of DNA will get 4C, double, same, no division. Okay. In metaphase, the content of number of chromosomes same, 46. Content of DNA 4C. But remember, in prophase, after anaphase, what happens in the anaphase? We will discuss. This dense chromosome will get separated. Are you getting my point? Their chromatid will get separated. You see here, we are where? We are in the prophase stage is finished, we are in the metaphase stage. In metaphase stage, you see, earlier it was like this one, chromosome, that is in the G1 phase or in the S phase. Are you getting my point? Earlier, chromosome has only one chromatid, but after DNA replication, it has formed another chromatid. This is another chromatid, two chromatids. Now it has two chromatids. This is the prophase, no? This is the prophase. We are now discussing metaphase. This is the metaphase. In metaphase, you see, there are two chromatids. How? We are talking about this after B, because this cell has undergone S phase, and in the S phase, the one chromatid has formed another chromatid because the content of DNA has increased. So these are the two chromatids. Chromosome is one only. Okay, M phase. Now they are loitering here and there in the M phase. I hope that you have understood what will happen in the M phase. These two centrioles in the prophase, we came to know that they are moving towards the pole. So these are the centrioles. It will form these fibers. This is known as the astral fibers. Astral fibers. Okay. These are also astral fibers. These are the two centrioles. These centrioles will produce what? They will produce some fibers, and these chromosomes having two chromatids, they will get attached in the centromere. They have the center, their center. You see, this is the one chromatid, this is another chromatid. Their center is known as centromere. Their center is known as what? Their center is known as centromere. So in the centromere, these fibers will connect. In the centromere, these fibers will connect, and all the chromosomes are aligned in the equator. In metaphase, all the chromosomes are aligned in the equator. Okay, so they are from metaphase plate. This is known as the metaphase plate. So in metaphase, what we observe, number one, if I write here, number one, metaphase plate is formed. Number two, chromosomes comes to equator. These are the two 3 points you need to remember in the metaphase. What happens in the metaphase? In metaphase, all the chromosomes are loitering here and there, as there is no nuclear membrane, but the centriole will produce acetyl fibers and some continuous and the discontinuous fibers. These are known as spindle fibers. These are known as spindle fibers. These are known as spindle fibers. With the help of the spindle fibers, they will attach with the centromere of the chromosome, and all the chromosome will be aligned in one plate. That means all the chromosome will be aligned in one. This is the equator. Okay, all the chromosome will be taken to the equator by the spindle fibers. Now, these spindle fibers can be of two types. One continuous, another one discontinuous. Discontinuous. Now, what are they? Let me see. You see, in continuous strand, these fibers, continuous strands, these fibers, you see, they are not attaching with the chromosomes. They are connecting direct from one pole to another. Those spindle fibers which are connecting directly from one pole to another, suppose this one, these are known as what? They are known as continuous fibers. Continuous fibers. Are getting my point? Or interzonal fibers. Interzonal fibers or continuous fibers. But there are some fibers which are connecting with the chromosomes. They have started from this pole, but they have not reached to that pole. They are connected with the centromere of these chromosomes. So these are known as the discontinuous fibers. Okay, the spindle fibers can be of two. I have told you this one. So what happens in the metaphase stage, I have told you. Now next stage, what is our next stage? Next stage is anaphase. So what will happen in the anaphase? You see, suppose this is a cell. So now you see these chromosomes, they have two chromatids. Now these two chromatids, okay, are bounded by the spindle fibers. Okay, are binded by the spindle fiber. These spindle fiber are now they are pulling this chromosome towards the poles. As a result, what will happen? You see, suppose this is one chromatid of one chromosome. This is a chromatid of one chromosome. They are going. Okay, they are going towards the poles. They are going towards the poles. They are going, this is the centromeres, and with their centromeres will be in the direction of the poles. So these are the centromeres. These are what? These are their centromere. So these centromeres are going towards the pole. This is the poles, centrioles. It has spindle fibers connected with the centromere of the chromosomes. It has spindle fiber. The centriole has a spindle fiber. These are the acetyl fibers. Okay. So now the spindle fibers connected with the centromere of the centromere of the chromatid. They are pulling they have separated the sister chromatids. This is the chromosome. This is the chromosome. This chromosome has two chromatids. Now the two chromatids are separated due to the pulling or due to the anaphasic movement. Due to the anaphasic, this movement of the chromatid, due to the splitting, it is also known as splitting. SPLITTING. Splitting of chromosomes. Okay, splitting of chromosome will take place in the metaphase stage. This is what? This is the metaphase stage. In the metaphase stage, anaphasic movement you will see splitting of the chromosome. Chromosome have been separated. Chromatids are moving towards the poles. Are getting my point? So these are the characteristics of the anaphase. Okay, the movement of the chromatid is known as the anaphasic movement. And you see, another thing you have to remember is the separation. Separation of what? What has taken place? Separation of chromosomes or separation of chromatid? I have told you this is a chromosome. This is a chromosome. This is a chromosome. It has two chromatids, sister chromatids. Now the sister chromatids are separated. So separation of sister chromatid. Separation of sister chromatid has taken place. Are you getting my point? Okay. Now these are all the characteristics of an anaphase. Okay. So now that's why I have told you. That's why you see in metaphase the number of actually this is a prophase. In prophase the number of chromosomes will be 46. Content of DNA will be 4C. Then metaphase I have told you it will remain the same, but in the anaphase. But in the same, as the two chromatids got separated. Okay, this is the metaphase. In metaphase, content of chromosome 46. Chromosome remains intact with two sister chromatids. Content within it intact 46. But you see in anaphase, I am sorry, this is not a metaphase. Sorry, it is an anaphase. No, this is anaphase. Okay. So in anaphase, in metaphase stage, the chromosome will come to the equator. In anaphase, the chromatid will get separated. As a result, the content of DNA will get half. content of DNA will get half. So here the number of chromosome is how much? 46 chromosomes remains will be same, but the content of DNA will get 2C. Number of chromosome 46 means in case of human being, it can for other organism we can say n. So it will be number of chromosome twice n, number of chromosome twice n, and content of DNA 2C. Now the content of DNA inside the chromosome, earlier in the prophase, in the metaphase, that chromosome were quite thicker because the content of DNA is double. But what happened here? How it became thicker? Like this, okay, like this, 2 chromatid. Now it is halved. In prophase it is 4C, content of DNA 4C. In metaphase it is 4C, content of DNA 4C. But in the anaphase, content of DNA will get half because sister chromatid were separated. So from 4C it will become 2C. From 4C it will become 2C. Okay. So next stage that we need to understand is the telophase. Now this is the telophase. Suppose you see this is the telophase. In telophase, all the chromatid have reached their poles. All the chromatids have reached their poles, south pole and north pole. These are the chromatids. These are the sister chromatids reach their poles. Okay, and they will lose their identity. Now the sister chromatids will lose their identity. Now they will in prophase what happened? From chromatin fiber they become condensed, they become thick, and they have taken the shape of a chromatid. But in the telophase what will happen? These are the centrioles. These are centrioles. No, these are centrioles. Okay. In telophase what will happen? You see, this chromatid will lose their identity and they will become thread-like structure like in prophase. They are attaining their ancestor stage. This is a chromatid fiber. Actually chromatin fiber. Yes. Okay. This is a chromatin fiber, thread-like structure. The chromatid lose their identity in the telophase, and now slowly the nuclear membrane will come to develop. Slowly the nuclear membrane will come to develop. So you see, from one nucleus, 2 nucleus has been produced. What I have told you? I have told you that we are discussing mitosis cell division, and mitosis cell division has two stage. Mitosis cell division has 2 stage. Mitosis cell division has 2 stage. One is a karyokinesis. Karyokinesis means division of the nucleus. So division of the nucleus is completed. Karyokinesis is completed. Are you getting my point? You see, from one nucleus, 2 nucleus has been produced. So karyokinesis is completed, but 2 cells are not produced. This is a 1 cell. This is a 1 cell only. So you see, in 1 cell, 1 nucleus should be present. Suppose in case of human being, in 1 cell, 1 nucleus should be present. That means cell is to nucleus. Cell is to nucleus ratio in case of human being. Suppose it should be 1:1. 1 cell contains 1 nucleus, but you see here. This is a 1 cell, nucleus is 2. Now cell ratio is not maintained. In this case, after karyokinesis, cell and nucleus ratio is not maintained. Cell is how much? 1. Cell remaining the same 1, but the nucleus is 2. So nucleocytoplasmic ratio. Cytoplasm is small, little bit area, but nucleus there are 2. Are you getting my point? So it needs to get divided. As the nucleocytoplasmic ratio is not maintained, it is not 1:1, it needs to get divided. That's why the cell will divide. I have told you. Now this cytoplasm will divide. Okay. Now cytoplasm division, karyokinesis completed. Now cytoplasmic division, it is another stage known as what you can say. Where I should write? Okay. Let me write here. No. Let me write here. Now cytokinesis will take place. You see, prophase, metaphase, anaphase. This is telophase. This is what? This is telophase. Now if you ask me, sir, what are the characteristics of telophase? In telophase, the chromatin fiber that is moving towards the poles, they have reached towards the poles, okay, and nuclear membrane will slowly gradually will appear, mitochondria will gradually appear, endoplasmic reticulum will gradually appear, okay, Golgi apparatus will gradually appear, all the cellular organelles will reappear, and the chromatid fiber reached in the poles, they have lost their identity, and what they have become chromatin fiber. These are the characteristics of telophase. Next stage is the cytokinesis, smaller one. See, in case of animal cells, suppose, let me write here, in case of animal cells, this is a cell will develop one furrow in the outside. This is known as what? This is known as a furrow, F-U-R-R-O-W, furrow, that is the invaginations. Okay, let me write here, no? Furrow, how it will divide? It will divide invaginations or foldings. Invagination in folding. Invaginations of plasma membrane. Furrow developed due to the invagination of the plasma membrane. Now this furrow will move towards the center. Now this furrow will move towards the center. This is the nucleus. This is the nucleus. This is the true nucleus. This is the chromatin fibers. These are chromatin fibers. So this furrow will gradually go to the center. And when they will meet, suppose when they will meet like this, when this furrow will go, when they will meet like this, from one cell, two cells are produced, you see. So this process is known as cytokinesis. Cytokinesis. That means the division of the cytoplasm. Nuclear division completed, but the division of the cytoplasm has not taken place. It will take place like this one in case of animal cell. But in case of plant cell, suppose in case of plant cell, this is a cell, this is a cell, this is the nucleus, this is the nucleus, this is a chromatin fiber. Okay, now the furrow will develop in the center. Furrow will be developed in the center, and this center is known as the cell plate. This center will form by the cell plate by one PHRAG. One structure is there which is known as the phragmoplast. Some protein will accumulate, some lipid will accumulate. They will combine with the phragmoplast and they will form what? Cell plate. This cell plate, in case of plant cells, okay, in case of plant, let me write here. In case of plant cells, cytokinesis will take place like this way. That is, it will move outwards. In case of animal, furrow is developed outer side. Furrow is developed outer side, and it has moved toward the center. But in case of the plant cells, not furrow actually, the cell plate will develop in the center with the help of the phragmoplast, and they will move towards the periphery, towards the boundary. And when they will reach the boundary, from one cell, two cell will be produced. Two cell division of the cytoplasm has taken place. Cytokinesis. In case of plant cell, division of the cytoplasm will take place with the help of by forming cell plate. In case of animal cell, division of the cytoplasm will take place with the development of furrow. Are you getting my point? So this is all about the mitosis. You see, in mitosis, that is the division of the karyokinesis. It is also known as the karyokinesis. First stage prophase, metaphase, anaphase, telophase, cytokinesis. Done. Are you getting my point? Cytokinesis means division of the cytoplasm. So mitokinesis is finished. If you ask me, sir, what is the importance of mitosis? From one cell, another cell is produced. Our body cell is produced. Okay? From one cell, millions of body cells are produced. In mitosis is also known as the chromosome number. Chromosome number remains same. It is known as the equational division. So when we started in prophase, are you getting my point? The chromosome number is twice n. Now here also in mitosis, from one cell we are getting two cell. From one cell we are getting two cell. Are you getting my point? The chromosome number will remain same. That is a twice n. Chromosome number will remain same. Twice n. The mitosis cell division is also known as the equational cell division. Okay? Because the chromosome number remains same. And from one cell we are getting two cell. And mitosis cell division will take place in diploid cells and haploid cells. Okay? From the diploid cell and haploid cell. What is its role? Its role is that it will help in the growth of the living organism, growth and the division of the cells, formation of the tissue, muscles, growth, okay, takes place with the help of the mitosis. Another one cell division is meiosis. Meiosis takes place in sexual reproduction for the formation of gametes. So this is all about the mitosis. This is the significance of the mitosis. See, mitosis also helps in the repairing of the dead tissues. Day by day we are forming new tissues, new cells are arising. New cells are arising only because due to the mitosis cell division. So I hope you have understood. Thank you.